Hey guys, so welcome back to the part 2 of chapter 1, which is unit 1.2, where we learn about the ultrastructure of cells. This is IB biology. So once again, it will be a very good idea for you to go and download the IB subject guides for any particular subject and have a look at what a particular chapter needs and wants of you and the breakdowns of the different units within that chapter itself. So for unit 1.2 of chapter 1, we will be exploring the essential idea that eukaryotes have a much more complex cell structure than prokaryotes. So the subject guide shown on the screen here will show you the nature of science, which links to your TOK that you need to know, which is tested in exams usually in your MCQ, usually worth 2 to 3 marks. Your understandings, which are concepts that you need to know that will potentially be tested in exams. Application skills and guidances, which are drawings, calculations, applications, skills that you need to know. For example, if you need to know a certain drawing, how do you draw it? And if the exam or the question requires you to draw a labeled version, what do you need to know and how do you need to know to label it? International mindedness, where you link the topic itself to a global, much more global setting. Theory of knowledge, as mentioned, and utilization, where you can see the links from this chapter and unit to other chapters and units across the subjects that you may study. So let's move on. So ultrastructure is basically all the structures of a biological specimen that are at least zero point 0.1 nanometers (nm) in their smallest dimension. So light microscopes, as you discussed in part 1, allow us to see the structure of cells, but very basic. You aren't able to see structures like ribosomes and mitochondria under the light microscope. But your electron microscope allows us to see ultrastructures of cells. They allow us to see them much more resolution, magnification also, and clarity for that matter. So there are limitations of light microscopes and electron microscopes that were discussed in part 1. So if you haven't checked that out, go and have a look. So, organelles. Organelles are basically specialized substructures within a cell itself that serve a specific function. So, prokaryotic cells, because they do not typically possess any membrane-bound organelles, but eukaryotic cells do. So, because of this difference, the functions and the survival mechanism and the reproductive mechanism of both prokaryotic and eukaryotic cells are very different. There are some organelles that they universally share, and as you learned in the in the previous video about endosymbiotic theory, you may know how things like chloroplasts in cells came about and how mitochondrion in cells came about as well. So go have a look again if you haven't. So prokaryotic cells and their features and functions. So prokaryotic cells are basically organisms whose cells lack a nucleus. They belong to the kingdom Monera, further classified into two distinct domains. So kingdom, phylum, class, order, genus, species. This is something that you learn later in chapter 5 as we go on. But for now, the two distinct domains are bacteria as well as Archaea. So Archaea is found in extreme environments like high temperatures or high salt concentrations or high pH of the environment itself, and hence these are known as extremophiles because they live in extreme conditions. Then we have eubacteria, which are traditional bacteria involving your E. coli, your Bacillus subtilis, and other known pathogenic forms which you can find in your digestive system and just anywhere else, in the air floating around you right now, you'll find E. coli. Features of prokaryotes, as mentioned, they do not have membrane bound organelles. So you have cytoplasm because obviously it's a cell, you need that cytoplasm. You have a nucleoid. This is interesting because nucleoid is basically a region where the cytoplasm, or region of the cytoplasm where the DNA is located. So you will have a DNA in that particular section, but you don't have a nucleus that encompasses that entire DNA and keeps it in, as we know formally. So they have a nucleoid instead of a nucleus. We have plasmids, ribosomes, cell membrane, cell wall, slime capsule, flagella, which allows movement, and pili, which may allow adherence to certain surfaces, mediate bacterial conjugation, and other features as well. So prokaryotes divide by binary fission, which is a method of asexual reproduction. So the the entire method is that the circular DNA is first copied in response to a replication signal. So because you are replicating it and you're going to form from one cell, it's going to divide to form two cells. You need to ensure that you have double the DNA material and double of everything else so that you can divide and divide equally. So you'll have to replicate the DNA. So the circular DNA will start to get copied and replicated, and then the two DNA loops will attach to the membrane, and the membrane elongates and pinches off. It forms a cleavage, as we know in cytokinesis, forming two cells, and hence that's how binary fission takes place. So as shown on the top right hand corner here, you will need to know how to draw prokaryotes. So here you have the cell wall, cell membrane here. This is the nucleoid, which is the region in the cytoplasm where your DNA is formed. These are your pili. You have your flagella. You have cytoplasm. You have ribosomes here, these little dots. So you need to know how to label and how to draw all of this. Of course, you can draw it in 2D. You don't need to know how to draw the 3D version of it. Here you have a typical structure of an animal cell. Of course, it doesn't have to be 3D once again, but you need to know how certain structures look like and how to label them. So once again, this is the entire mechanism showing how binary fission in a prokaryotic cell takes place. So here you have the replication of the DNA material. Here your DNA material is attaching itself to the membrane. Here the cytokinesis, which is a pinching off, which is taking place, and then you have two cells in the end. So binary fission. So this is the drawing and the labeling of the prokaryotic cell. Take note so that you can know how to draw it. All right, so prokaryotic versus eukaryotic cells, as I mentioned earlier before, they have a lot of differences, and that results in their functions and their survival capability and mechanism, reproduction, all of them being different. So you can remember this through a mnemonic, which is known as DORA. So that's DNA, organelles, reproduction, and average size. These are the four main umbrella groups of differences between prokaryotic and eukaryotic cells. So prokaryotes, their DNA is naked. You call it naked because it's not encompassed in a certain membranes. It's just floating around in that certain region of the cytoplasm. The DNA is circular, and there are no introns. You will learn this later on in um, chapter 3 and 10, where you learn about DNA transcription translation, what introns is. But introns are basically the useless parts of your genetic material that does not get transcribed into proteins. So you'll learn about this later on. But if you're confused right now, it's okay. Eukaryotes are basically, in terms of DNA, they have DNA bound to a protein. So that protein is known as a histoprotein, and you'll have eight proteins around that particular DNA. So it forms an octamer. You will learn this later on in the chapter as well. The DNA is linear as compared to the circular ones in prokaryotes, and they have introns in eukaryotes. Now, in terms of organelles, prokaryotes, as mentioned again, they do not have a nucleus while eukaryotes do. They have no membrane bound structures, but eukaryotes do. And prokaryotes have 70S ribosomes while eukaryotes have 80S ribosomes. So this is something once again that you will learn in chapter 3 and 10 when we learn transcription and translation, the significance of 70S and 80S ribosomes. So for now, all you need to know is the difference in the type of ribosome that are there in two kinds of cells. Reproduction, like I mentioned earlier on, prokaryotes do it by binary fission, while eukaryotes may do it through mitosis, meiosis, and in prokaryotes, you only have a single chromosome, which is haploid in nature. That means it only has n number of chromosomes, while your eukaryotes, your chromosomes are paired up, so they can be diploid or even more. Average size, in terms of size, your prokaryotes are much, much smaller, while your eukaryotes are much, much larger. So this is a overlook of the entire of eukaryotic organelles. So you have your endoplasmic reticulum, you have your smooth RER and your rough RER. Smooth RER is not studied in IB, so you only need to know it exists, but you don't need to know its function and stuff. You will need to know about RER though. You have the mitochondria, you have the centromeres, centrosomes, you have nucleus, Golgi apparatus, proxysomes, and in plant cells only you will have chloroplasts. In addition to the ones shown in the previous slide, you will also have chloroplasts, vacuoles, cell walls, and in animal cells only you will have lysosomes. So, in detail, let's go in. So, nucleus, it's generally spherical in shape, and it is a double membraned organelle. So, as we go on, you will realize that certain organelles are double membraned, while certain are only single membraned. You will need to know which are double membraned and which are only single membrane. So, things like nucleus, mitochondrion, chloroplast, these are all double membrane. So nucleus double membrane, it has pores and holes. You will learn the significance of this later again in chapter 3 and 10. This is where your transcription material comes out from the nucleus so that your mRNA and can be copied and transcribed into proteins. So those holes are needed in your nucleus. The nucleus also contains the genetic information in the form of chromosomes, where your DNA, as mentioned earlier, DNA and associated histone proteins are present. In the nucleus itself, the chromosomes are uncoiled and they are referred to as chromatin. They stain a dark color and are concentrated at the edges of the nucleus. As I mentioned in chapter 3 and 10, you will learn where mRNA is transcribed in the nucleus and then it leaves the nucleus via the pores. Because DNA is too large to move, you need to copy it into mRNA, and that single strand of mRNA then moves out. So now moving on to free ribosomes. In eukaryotes, as mentioned, you have 80S ribosomes as compared to the 70S ones in prokaryotes. They are larger in size. They have no membrane. They appear as dark granules in the cytoplasm, and they help to synthesize proteins to function in the cytoplasm for use within the cell. So you have free ribosomes in the cell, and you also have those ribosomes which are attached to your RER, which make the whole thing look as a rough structure. So free ribosomes synthesize proteins to be used within the cell, whereas ribosomes on your RER, which is your rough endoplasmic reticulum, synthesizes proteins to be transported outside the cell. So those proteins will be used outside the cell by other parts of the body. So you need to know that difference as well. Mitochondria or mitochondria is a plural version of mitochondrion. In IB, they are very specific about plural and singular forms of the word, so do take note, because sometimes if the question asks you for only a single mitochondrion and you write mitochondria, you may lose a mark. So it's just safer to know both the versions. So mitochondrion, as seen on your right hand corner, you again need to know how to draw and label a mitochondrion. So it is a double membraned organelle, as I mentioned earlier as well. It has a smooth outer membrane and a folded inner membrane, and the folds, as I'm pointing with my cursor, are known as cristae. They vary in shape, and the size of the the site of the ATP production by aerobic respiration is your mitochondrion is basically the site where ATP is produced. So ATP production is something that you will learn in chapter 2 and 8 when you learn about aerobic respiration in plants and animals. So we'll cover that later on, but that's where the common saying comes from, that mitochondrion is the powerhouse of the cell. But you will learn greater details about the powerhouse of the cell in later chapters. When you have your rough endoplasmic reticulum, your RER, as I mentioned, you do not study your smooth endoplasmic reticulum structure in IB, but you do need to know how to draw your RER and label it um, in the cell diagrams when asked for. So it consists of a flattened membrane sac. They are often located near the nucleus. ATS ribosomes are attached to the outside of the cristae, and they are ribosomes. And your RER synthesizes proteins which are transported by vesicles to the Golgi apparatus for modification before they are secreted and used outside the cell. Microtubules, they are basically small cylindrical fibers which have a variety of functions. For example, they can be part of the flagella or they can play a role in cell division. Centrioles consist of two groups of nine triple microtubules. They are found mainly in animal cells, not so much in vascular plants or fungi. Vesicles, as mentioned earlier, they are a single membrane with fluid in size. They are very small in size, and they are a transporter. So they are used to transport materials inside a cell. As mentioned in the previous slide, your RER uses it to transport the protein that it synthesizes to the Golgi apparatus. So now the Golgi apparatus is an organelle which also consists of flattened membranes sacs called cristae, like your RER. But they are different because they have no attached ribosomes, and they are often sited closer to the plasma membrane, whereas your RER is closer to the nucleus. Your cristae are shorter and they are more curved than those of the RER. And your Golgi apparatus's main function is to process or modify proteins from the RER, and proteins are then repackaged in vesicles for secretion outside the cell. Macules, these are found in your plant cells, uh, sometimes in animal cells, but in animal cells they're more smaller and temporary, whereas in your plant cells, they are much huger, much bigger, and they're more visibly present. So vacuoles are also single membraned with fluid inside of them. In plant cell, their majority function is to ensure that they maintain the cell volume and the turgor of the cell so that the cell does not lyse and collapse. Whereas in animal cells, they're much smaller and temporary, and they're used for various reasons such as to absorb food and digest it. Lysosomes, as mentioned, these are only present in animal cells. They are generally spherical with a single membrane, and they are formed from Golgi vessels. So they contain digestive enzymes for the breakdown of ingested food in vesicles, unwanted or damaged organelles, the cell itself, and also high concentration of enzymes that cause the organelle to stain heavily and hence appear dark on micrographs. So micrographs are basically diagrams and pictures that you can take when you're looking at that organelle using a microscope. So using a microscope, you get a micrograph. Cilia, again, once again, it's only in animal cells. They are basically thin projections from the cell surface. They contain microtubules and are used to either move cell or move fluids next to another cell. So, flagellum, as I mentioned this earlier when we were talking about prokaryotes, or flagella, if you're talking about many flagellum, so the plural form, they are again only in animal cells. They are also thin projections, usually singular, from the cell surface, and they contain microtubules and are used to move the cell. So cell wall, again, this is only in plant cells. This is something that's already been covered in secondary school, so I won't touch too much on it. But do know that it is mainly made up of cellulose. And then you also have your chloroplast. Take note that this is a double membraned organelle. And we will learn more about the structure and the shape of chloroplasts as we go on in chapter 9, where we learn about plants. So this again is a small table where you can see the overall comparison of plant and animal cells. Do have a look. It's a more condensed version and easier to understand if you're looking for a quick reference. All right. So as part of this chapter, earlier in the first part of the chapter, unit 1.1, we learned about light microscopes. So now in unit 1.2, we have to learn about electron microscopes. So electron microscopes, as the name itself says, they use electron beams focused by electromagnets to magnify and resolve microscopic specimens. So in a light microscope, they use light in order to shine on that specimen and then bring to life that specimen so that you can have a look what that specimen is. Whereas in an electron microscope, you shoot electrons using electron beams and electromagnets in order to magnify and resolve microscopic specimens. So resolution, once again, do know the definition because it's important and it does come out during exams. So as mentioned, I think in unit 1.1 we did talk about TEM and SEM, your two types of electron microscopes, transmission and scanning electron microscopes. So TEM generates high resolutions of cross sections of objects, whereas your SEM can be used to display enhanced depth to map out the 3D surface of that particular object. So the advantages and disadvantages were discussed earlier in the first part of the chapter during unit 1.1, but here's a summary once again. So cell micrographs, as I mentioned, micrographs are basically what you obtain once you are using a microscope. So micrographs are basically photo or digital images taken through a microscope to show a magnified image of a specimen. So while organelles have identifying structures, specific shapes may vary depending on the location of the cross sections. So attempts can be made to deduce the cell function based on the relative abundance sometimes of various organelles such as mitochondria. So if you have more mitochondrion in a certain cell, you can maybe deduce that this cell does a lot does a lot of aerobic respiration activity, and hence is maybe used in something like muscle cells, because muscle cells, you do need a lot of energy to move, contract, relax. So that if you do have a lot of chloroplasts, you know it comes from a plant cell. If you have a lot of RER, that means your cell is involved in production of large amounts of proteins that are transported out of the cell. So it is very important that you have your concepts tight and straight, so that you know in a question when and what can be applied. So remember, IB, it's not difficult, it's just tricky. You just need to know your content, and you need to be able to see the larger picture when you do your questions. All right, so eukaryotic structures of your animal cell. This is something I mentioned already, and I went through the structures as well. Do know that you need to know how to draw an animal cell in 2D and label it as well. So the drawing of it is on your right hand corner of the screen. Do have a look and try drawing it out, and practice it a few times so that you get used to how to draw it. And then if asked in the exam, you can do it much easily. Plant cells as well, this is something that you have covered earlier in education. You should be able to know how to draw a plant cell by now, but for reference, it is here again on the right side of your screen. I have already gone through with you the organelles as well, so do practice how to draw it, so that once again, if it does come out during the exams, you know what you need to do. All right, and so that is the end of today's video. We went through the second part, which is unit 1.2 of cell biology, and I hope this video helps. So I'll be coming out with more parts to the rest of the chapter, and I hope this helped. Thank you.